Back friends, welcome back in another video tutorial from microbiology with Shagnik. So in that video tutorial we are going to discuss about ultracentrifugation. This video will going to be a video where you will get everything that you need to know from ultracentrifugation. Okay, so let's discuss about everything one by one. So first of all, what is ultracentrifugation? So ultracentrifugation is nothing but a simple centrifugation. First thing is that, but it is more sophisticated and advanced version of the instrument, and that can control or that can operate a extremely high speed. Okay, so what is the utility of that high speed? This high speed will help you to separate molecule that you cannot separate with the help of just a normal centrifugation. That is why named ultracentrifugation. Okay, that means a high power centrifugation. Okay, that operates at extremely high speed, near about a speed of 60,000 rpm to 1,50,000 rpm. So here I have provided some basic information that the speed of the ultracentrifugation, and remember they are usually larger in size, and in the ultracentrifugation you can maintain your sample either in batches or in continuous flow system. Okay, so most of the ultracentrifugation are refrigerated to control the heat generated by the speed of the rotor. So what is the need of that? So remember that we are using ultracentrifugation for separation purpose, right? What kind of separation purpose? Biological samples separation purpose, right? So in biological sample we have DNA, RNA that can be degraded or denatured with the help of heat. That is why we need to control the heat by appropriate refrigerator system. Otherwise our result will be altered. That we need to keep in our mind. So up to that point we have learned that ultracentrifugation is nothing new. It is a simple centrifugation process where the difference is it operates at extremely high speed and allow us to separate samples that cannot be separated with the help of just centrifugation. Okay, and we have also learned that what is the utility of using a refrigerator for ultracentrifugation, that it will control the heat that can create a error in our measurement. Okay, so now let's discuss about the principle. Although I am not going to discuss about principle in such a way, in a descriptive way, I am going to take the help of a diagram so that you can easily understand. So let's move to the diagram. Okay, so welcome again. Now first of all let's learn about what kind of abbreviation I have used here so that you can easily understand what is the principle. So here I have used a X star and X. Remember, X star and X are the compound of same molecular weight. Okay, but X star is denser than the X, and I have used Y star and Y where the condition is exactly the same. Y star is denser than the Y and they are of same molecular weight. Okay, and here we have taken a mixture of 4 particles, X, X star, Y, Y star. Okay, so a ultracentrifugation, remember, works on normal centrifugation procedure. That means the same thing that I have discussed in our centrifugation principle discussion video. It is based on the sedimentation principle. Okay, so what is the sedimentation principle meaning? Sedimentation principle means the denser particle will be settled down faster than the less dense one under the influence of the gravity. That is the basic principle in which the sedimentation our centrifugation force our centrifugation principle rely on. So now my question is, if it is rely on the gravitational force, then what is the need of a centrifugation machine? Because we can simply keep our tube with our desired sample, then we will just look for the separation, then it is done. Then what is the need of the centrifugation machine? So remember that the biological molecules we are handling with the help of that centrifugation procedure are too small that we cannot separate them just by with the help of gravitational force, because in case of two smaller molecule there is a random thermal motion that prevents separation, okay, or more precisely saying that random thermal motion prevent those kind of molecule from pillating, okay. Not only that, the separation is possible, yes, that is true, but it takes a lots of time, okay. So if you have the time, that much of time to invest, then you can, but what is the utility of this, right? So for getting the result of our sample within a very small time, we have developed centrifugation machine, where the tube is just rotated at a speed, certain speed. That is the idea. And difference between centrifugation and ultracentrifugation is, ultracentrifugation can generate more speed than in case of centrifugation, normal centrifugation, okay. So now the question is, what is the utility of that speed? So remember, during that rotation, each of the molecule will also rotated about an axis, and this rotation result in the formation of a perpendicular force that is acting on every particles on the sample, and this force helps the gravitational force to perform the sedimentation. This force is known as centrifugation force, and that is also acting on downwards, okay. So under the influence of the centrifugal force, larger molecule will move faster than the smaller one. Let's consider the y is larger than the x, then y will move and settle faster than the x, and the denser particle will be moved towards the periphery, outwards the periphery of the tube, okay, and smaller less dense particle will be moved towards the center of the tube, okay. That means denser particle will rotate outwards the periphery of the tube, and less dense particle will rotate towards the center of the tube, okay. That is the reason why after sedimentation you will get larger and denser particle as a pellet, and smaller and less dense one will remain floating on the supernatant, okay. So that is the principle of the normal centrifugation as well as ultracentrifugation. Funda is exactly the same. Here we are generating centrifugal force by spinning the tube in a centrifugation machine, okay. That helps us the gravitational force to sediment our particles. That is the idea. Larger and denser particle will sediment or settle down faster than the smaller one, and that is how the separation is done, okay. So good. So remember now that this ultracentrifugation, I am denoting this ultracentrifugation as UC, can be divided into two. That is, one is AUC, another one is PC. AUC stands for Analytical Ultracentrifugation, and PC stands for Preparative Ultracentrifugation. So Analytical ultracentrifugation, if you have watched the very first video I have uploaded regarding centrifugation, then I hope you know what is analytical centrifugation, although I am going to discuss about analytical centrifugation here too. So analytical ultracentrifugation or analytical centrifugation means with the help of that centrifugation, analysis of each of the particles in the sample mixture is done. Okay, that means here we are analyzing the particles in the sample. What is the molecular mass of them, what are the hydrodynamic properties of them, okay, and what are the size, shape of them. That means the physical property determination can be done with the help of that method. So let's see what is analytical ultracentrifugation. Okay, so analytical ultracentrifugation, as its name suggests, is a technique that can analyze various particles present in the sample. Okay, so analytical ultracentrifugation employs a detection system that monitors the spinning and the position of the particle. Okay, so now you may ask what are the utility of this, and this detection will be in real time, that means when our centrifugation is going on. So you may ask that what is the utility of knowing the position and the spinning, because the utility is it will help you to determine the sedimentation coefficient, okay, and that in turn help you to analyze the macromolecule. Okay, so sedimentation coefficient we have discussed earlier, that V by omega square r. Rate of it is the ratio of the rate of centrifugation by angular acceleration, omega square r. So sedimentation coefficient that I am talking about is V by omega square r. V is the velocity, also known as rate, and omega square r is the centrifugal acceleration. So with the help of that ratio we can determine the physical properties of that macromolecule, and in analytical ultracentrifugation we can determine it real time. Okay, so relative molecular mass determination can be done with the help of sedimentation velocity approach and sedimentation equilibrium methodology, although we are not going to discuss about those things in details. Hydrodynamic property of macromolecule can be observed with the help of that same principle, sedimentation coefficient, that means again V by omega square r. Okay, so sedimentation coefficient also characterizes the change in the size and shape of the macromolecule by changing the experimental condition. Okay, that means how the size and shape changes with experimental condition, that means we are using different kind of rotors, we are changing the speed, etc. So sedimentation coefficient can help us to characterize the change of size and shape during changes in experimental condition, and three optical systems are available for detection: absorbance, interference and fluorescence, that give us the precise and specific idea about sedimentation in real time. Okay, so that is the idea. That means here the main goal is to detect the position and the spinning real time during the centrifugation, that help us in determining the sedimentation coefficient, and with the help of that sedimentation coefficient we can analyze the properties of them. Okay, for example, Schlierian optical system is, for example, where we are observing the particles in real time to determine the position and movement. Okay, and this position will give us a idea about sedimentation coefficient. Okay, and this sedimentation coefficient in turn help us to analyze the particle. That is how easy analytical ultracentrifugation is. So with the help of that analytical centrifugation we can analyze biological samples, for example our DNA, RNA, proteins. Okay, so in analytical ultracentrifugation, as its name suggests, we are analyzing the particles present in the sample, right? So for that purpose we are employed a detection system that detects the presence of particles and spinning live during centrifugation. Okay, and this data will help us to determine what is the sedimentation coefficient, and sedimentation coefficient help us to analyze what is the properties of those macromolecules. Okay, for example, relative molecular mass determination can be done with the help of sedimentation equilibrium methodology or sedimentation velocity approach. Hydrodynamic property of the macromolecule can be determined with the help of sedimentation methodology, sedimentation coefficient. Sedimentation coefficient can also help us to characterize the changes in the size and shape of the molecule, okay, with changes in experimental condition. And various kind of optical systems such as absorbance, interference and fluorescence we can use to precisely determine what is the sedimentation in real time. Okay, so for example, we have seen the Schlieren optical system where we can determine the particles position and movement in real time, and with the help of the data of position we can determine the sedimentation coefficient, and sedimentation coefficient help us to determine what is the hydrodynamic property of the particle, what is the analytical property of the particle. That's how easy analytical ultracentrifugation is. Now let's move to the next one that is quite easy than analytical ultracentrifugation, that is preparative ultracentrifugation. Okay, and as its name suggests, preparative ultracentrifugation help us to separate or isolate particles from a mixture. Okay, in preparative centrifugation, remember very carefully that contents are analyzed after centrifugation, unlike AUC where we are analyzing our contents during centrifugation with the help of those detection system, optical systems, right? That is the very important point. It is required for writing the difference between analytical ultracentrifugation and preparative centrifugation. These 2 points is very important. And preparative centrifugation, remember, separate particles on the basis of density and their sizes. Okay, and that is why we can get a idea about density and size of the particle too. That is quite analytical type of centrifugation too, right? So that PC can be operated for density gradient isopycnic centrifugation and differential centrifugation. Okay, so density gradient and isopycnic centrifugation, particles of the sample are separated on the basis of their density. Okay, so in the test tube different particles of the sample are separated, okay, and the samples are isolated in the form of bands in distinct level. Okay, and in those positions the density of the samples equals the density of the medium. Okay, so our preparative ultracentrifugation can be operated with density gradient centrifugation and isopycnic centrifugation where particles are separated on the basis of their density. Here the particles can be separated at distinct level and they can be isolated in distinct band. Okay, where the density of the particles, each of the particles that are present in that band are equal to the density of the medium. On the other hand, the differential centrifugation, where our particles are separated by applying different speeds, okay, by different speed of the rotor. I have discussed about differential centrifugation in separate video where I have explained that how by increasing the speed of the rotor we can separate the smaller molecule, right? A larger molecule will demand a smaller speed and the smaller molecule will demand a larger speed. Okay, so preparative centrifugation we have learned help us to isolate and separate macromolecules or a desired molecules from a mixture of sample. Okay, So it can separate molecules on the basis of density and size. Preparative ultracentrifugation separate on the basis of density when they are operated for density gradient centrifugation and isopycnic centrifugation. Okay, in those cases they are separated on the basis of density, and in case of size the separation is going on when they are in differential centrifugation, right, where by generating or by increasing speed we can separate more larger molecule, more smaller molecule. Okay, larger molecule will demand less speed than the smaller molecule. Okay, so that is the two kind of centrifugation, ultracentrifugation procedure. One is our AUC, that is analytical ultracentrifugation, that requires analysis of the samples real time. Another one is PC, that separates the macromolecules from the mixture. Okay, and this is the principle of the ultracentrifugation that is based on the general principle of the PC. That is based on the general principle of centrifugation. Okay, so now let's look our last topic, that is the use of ultracentrifugation. Although we can make out the use here too. So preparative centrifugation is used for purification of biological crude extract on the basis of their density and size. Analytical ultracentrifugation can help us the analysis of biological molecules such as DNA, RNA. Okay, and ultracentrifugation most importantly separate mixture of sample that our centrifugation cannot. That is the very important line for ultracentrifugation. Okay, so now I am just displaying the slide so that you can take screenshot from that for your notes. So here everything is described what I have described diagrammatically. This is how the centrifugal force generated due to the rotation of samples about an axis, perpendicular force, centrifugal force generated, which is acting on every sample and helps sedimentation our gravitational force to sediment the particles. Okay, analytical ultracentrifugation, very important one, that provide us the data of sedimentation coefficient real time. And then we have discussed about the preparative ultracentrifugation, that is also an important one. Okay, then the preparative centrifugation is used for in biology for pillating fraction of organelles like ribosome, even viruses, depending on their density and size. And ultracentrifugation used for determination of densities of various macromolecules we can done with the help of PC, and it is also used for purification of biological crude extract. And analytical ultracentrifugation determines properties of biomolecule like protein and nucleic acid. So this is the basic concept of ultracentrifugation. So I hope you have enjoyed the video well and understood it well also. So thank you for listening to this class.